Are we ready? Howling, feedback. All right. Hello. All right. Okay, everyone, let's go ahead and get started. Welcome to the session, RMB, rhythm, beat, and meter processing. Okay, so I'll be chairing this session, and I'll be the first speaker as well. So I guess we'll still have more audience here. Let me go ahead and get started. Okay. All right. So I'd like to ask you to raise your hand, whether left or right. And now I'm going to have you do tapping. Okay, move your finger, index finger, for the next 10 seconds. Okay, ready, set, go. Okay, that's good. So I see that some people tend to move faster than others. Okay, now, how many of you would believe if I say I can predict your memory skill by measuring your tapping interval? In other words, your finger tapping rate. Raise your hand again. Not many people. Some, few, right? Yeah, I agree with you. Seemingly very unrelated. In fact, if I do literature search, there's not much out there. So if you type in keyword, working memory, finger tapping, and so forth, there's not much. But here's one of the few. So what you see here is the multi-section slice view, showing the slice of the cerebellum area. For working memory, this column, and motor rehearsal, and finger tapping. And you can see that some areas in the cerebellum, there's some overlap between working memory and finger tapping, right? And here's a table from one study about working memory deficit in schizophrenic patients. Okay? And as highlighted by this red box right there, the finger tapping, I don't know, the font size may be a little too small, but the finger tapping task is positively correlated with the backward working memory. So there's significant correlation. And that's pretty much it. Now, for the next several minutes, I'm going to illustrate some neurobiological background that may suggest the connection between rhythm and working memory. Okay, so we'll be revisiting this slide later on, but just bear that in mind. Okay, so here's a little bit of biology and neuroanatomy. Okay, so what you see here is the coronal section of the brain. Okay, here different colors denote different types of a dopamine receptor. So here green color means D1 to D5, and blue and red. Now I'm going to highlight this portion called the basal ganglia, where there's a lot of dopamine D1 and D2 receptor. Okay, as you see here, it's called the striatum basal ganglia. And on the right side, we have a cartoon of dopamine receptor. Okay, so depending on this G protein, inhibitory G protein type, that determines the dopamine subtype. Okay, so that's cleavage where the dopamine is binding, right? Now there's a gene called the DRD2 gene. Okay, and some people are born with slight mutation in that gene. So one codon is swapped. Okay, so it's called SMP, or single nucleotide polymorphism, or SNP. Okay, now the consequence of that mutation is the reduction of D2 receptor in the striatum. Okay, and that caused some slow sensory motor processing, right? So I'll show you in a minute. Those people tend to move their fingers rather slowly, and also, quite interestingly, this DRD2 gene is associated with a poor grammar ability. Okay, and also this DRD gene polymorphism is associated with a poor working memory. In fact, there is quite a bit of studies out there. So these are some of those studies titled. Okay, so DRD2 gene modulate the effect of ventral striatum activation on working memory performance, and so forth. Okay, so they all came out in pretty significant journals. Now, this is a guy named Martin Wiener. We used to be an office buddy when we were both postdocs at UPenn. And this is the guy who got me into this working memory and rhythm. So I'm going to illustrate his study briefly. Okay? So here what you see is the finger tapping, right? So what he did was he recruited Penn undergrad students and had them move their fingers. He didn't ask them to move fast or slowly, just at their own tempo. Okay? And then after that, he genotyped on a couple of genes, including DRD2 gene and COMT gene and BDNF gene. They're expected to be sort of candidate genes for this motor processing. Now, what happened is that people with DRD2 polymorphism, which is denoted by A1+, meaning that they're the A1A carrier, right? That's a DRD2 mutation. Those people move their fingers slowly. And this interval difference, the y-axis is the mean inner tapping interval. They tend to move their fingers rather slowly without any influence whatsoever. So that suggests that somehow their motor processing or temporal processing is inherently slow or compromised. So he and I teamed up for the follow-up study. It's a neurogenetic fMRI study. So just like his behavioral study, we genotyped the subjects, and then we put them in the magnet, and we gave them some temporal judgment tasks. And I'm not going to go through all the nitty-gritty details about this study, but here the upshot is that those people with DRD2 polymorphism, they tend to activate the basal ganglia more strongly when the task is harder. So here the sub-second task is harder than super-second temporal judgment, and you can see that there's a significant differences. Whereas in the control, who don't have any DRD2 polymorphism, they don't show such differences. So something's happening in the basal ganglia in those DRD2 polymorphism group. Now, so here the question is, then does rhythm tapping interval predict working memory, right? That's the question that I asked earlier on. Now, so here the main hypothesis is that because rhythm tapping and working memory is supposed to be dependent on this temporal processing, right? If we measure someone's rhythm tapping rate or their variability, we can predict working memory and vice versa. Okay, so that's sort of the model that we are testing. Okay, so again, if people have a relatively faster temporal processing or efficient temporal processing, they will manifest as faster finger tapping in the motor domain or better working memory skill in the cognitive domain. So that's the model that we're testing out. So this is the picture of a COSI, the Children's Museum in Columbus, Ohio. Okay, and on the second floor we have a lab space called the Buckeye Language Network Language Pod. So here are some of my students there. So what we do is we reach out to parents and kids and ask if they're interested in participating in our study. And if they say yes, then we run them on some of the tasks, okay? Some memory tasks, some rhythm tasks. So altogether, the entire procedure has to be done in 20 to 25 minutes, okay? So in this study, we have a total of 81 typically developing children. So we submitted the abstract that was 85. Later on, we found out that 4 subjects had to be discarded. They didn't meet the criteria. So down to 81, but it didn't change our overall result and data at all. And there are 47 females, and overall age range is 11 years old, plus minus 2.5 years. And these are the measures that we obtained. Okay, so we measured their language background, whether they're monolingual, bilingual, or trilingual, their SES, socioeconomic status. As a proxy, we measure mother's education level. We also gave them auditory and visual working memory. I'm going to show you some example. And just like what you guys did, we had kids do their self-paced drum tapping. And also, we measured their rhythm synchronization test. I'm going to show you the slide of example 2 with 3 different BPMs. Okay, and we analyzed this data using logistic regression within linear mixed effect modeling framework in R. Okay, and we tried to predict auditory and visual working memory separately with all these measures. Okay, and we also performed some parity tests and correlation as well. Okay, so here's an example of auditory working memory. Okay, let's just watch. Okay, you got the idea, right? So if you hear this sound, yes, left, no, right. Yeah, pretty easy, right? So who said yes? Who said no? Nobody. Wow, you guys all got it right. Thanks for the attention. And here's the example of a visual working memory. All right. Say yes or no? Yes? Who said no? There are a few no's. Okay. It's harder, right? Yeah, it's actually harder. Kids found it harder. I'll show you the result in a second. And this is an example of a rhythm synchronization. Yeah, it's from one of the, it's a 120 BPM. So we developed this simple app and we install it in the computer. Kids just hit the keyboard in response to metronome sound. Okay. All right, here's the result. So as you just heard, visual working memory is a lot harder than auditory working memory for kids. There are significant differences. But for rhythm synchronization, overall, kids did a fairly good job. They're all above around 90% accuracy. And this is the result of some plots from LME, the logistic linear mixed effect modeling plots. So here on the top left corner, we have main effect of age. So on the x-axis, we have ears, age. On the y-axis, is auditory working memory accuracy and throughout, okay, all the y-axis. And not surprisingly, right, kids who are older, they outperform younger kids. So there's significant main effect of age. And in the middle here, so this is sort of the core key finding we had. On the x-axis, we got this normalized mean intertapping interval, right? So we standardized the intertapping interval, right? And y-axis again, so auditory working memory score. So as you can see, the longer the mean intertapping interval, the lower the auditory accuracy, working memory, right? In other words, kids who tend to move their finger faster, they have a higher auditory working memory score. And we also looked at the variability. It came out significant, but in somewhat opposite direction. So kids with higher variability of this finger tapping, they tend to have a higher auditory working memory score. We tried to make sense of it, but what we found is that kids who move their finger faster, they also have a bigger variance, standard deviation. So there is a really strong correlation. But both explain the auditory working memory. And unlike a rhythm tapping interval, this rhythm synchrony actually didn't predict their auditory working memory score at all. And we also looked at the visual working memory score, given that both are on the working memory domain, but visual working memory didn't predict their auditory working memory performance. And there's no difference in sex. Okay? And here's the result of a visual working memory. So just like auditory working memory, older kids tend to outperform younger kids, right? And for some reason, we still have tried to make sense of this. Boys tend to outperform girls in visual working memory tasks. It was not the case in the original 85, but after we discarded those 4 subjects, came out significant. We tried to understand, interpret this data. Anyhow, and also the auditory working memory didn't predict, just like a visual working memory didn't predict the auditory. And none of these rhythm synchrony, tapping interval, or tapping interval, mean of the tapping interval, or standard deviation of a tapping interval, they didn't predict visual working memory. Although, if you look at it, there's a trend, right? Kids with a longer mean-to tapping interval, their visual working memory score is relatively lower than the other kids, but it didn't reach the significance. Okay. So, so altogether, here's some discussion points. So we found that the rhythm tapping predict auditory working memory performance. So this is the model that we were testing, because rhythm tapping and working memory, they're all dependent on temporal processing. If you can measure their rhythm tapping interval, then you can actually predict the working memory as well. And we found that that was actually the case. So unlike a rhythm tapping interval, rhythm synchronization did not predict auditory working memory. So this is actually consistent with the previous report, where they suggested there are different multiple rhythm skills. Okay, so there's skill for beat tapping, there's skill for memory sequencing, and here on the right side, on the x-axis we've got metronome tapping, on the y-axis tapping along to some music sequence, so it's a free tapping, and there's no correlation. So our result, our data is consistent with what they found. We also found there's no correlation between auditory and visual working memory. So I've been told over and over again that it doesn't matter whether you use auditory or visual working memory when it comes down to working memory measure, but as it turns out, it's actually very specific to sensory domain. So if you were to study auditory phenomena, then you have to use auditory working memory, not visual working memory. And that's because we use, as I described earlier, unnameable objects, right? So you're hard to name them. And when you use those, then there's no correlation. But if we use something like animal sounds, right, tiger, and they all mapped onto some semantics, then it probably wouldn't matter whether you use sound or pictures because they all mapped onto this semantics. But when you cut that sort of a semantic mediation, then you no longer see correlation. So here we've got correlation of 0.1, which is not significant. So that's another finding we have. So given all this connection, the neurogenetic and behavioral connection between working memory and rhythm, it's reasonable to ask, can music training help improving working memory? And the answer will be, as you can guess, yes, right? There are some studies out there. So here's one of those. So this study showed that the main effect of music training on several various cognitive skills, including visual spatial working memory and the verbal working memory. So this music training group actually outperformed non-music training group. And this is a neural data, EEG data, where they show this sort of a pronounced P300 component in response to deviant in the working memory task involving oddball paradigm. So when musicians listen to oddball stimuli, they develop stronger activity in the P300 signature. Okay. All right. So these are 4 take-homes here. So I've shown you that rhythm tapping predict auditory working memory performance, but rhythm synchronization did not predict auditory working memory. There's no correlation between auditory and visual working memory, and music training can help improving working memory skills. So altogether, I've shown you this slide early on. So we're studying this model, right? So there's this neurobiological foundation that provides connection between rhythm and working memory. Okay, so they all depend on temporal processing, and this temporal processing is mediated by this dopaminergic system, mainly by frontal striatal pathway. And there's this genetic influence, such as DRD2 gene, right? If you have a mutation in that gene, then down the road you're going to have impaired rhythm or working memory that manifests itself as in the motor domain or cognitive domain. All right, so I want to thank my student, Kate Corbeil, who just graduated. So that was her undergrad project, and she just finished this, and unfortunately she can't make it today because of her summer job. I really want to give a big thanks. And these are the students in the lab who were involved in this project. Thank you for your attention. Two questions. So I'll get 2 questions. All right. Huh? In this room, and then 2 more. Okay. 2 from in this room, 2 from Columbia.
Very good. The question I have is about the basic spontaneous motor tempo measurement. This was just a one-shot experiment, a one session. The kids came in, you did the tapping, and got their baseline SMT on one observation, correct?
Yes, given the time constraint.
Yeah. I've, as a lark for when I teach music perception cognition, I actually have the whole class measure their spontaneous motor tempo, and I have them do it every Saturday morning for 6 weeks. And a lot of people are very consistent, but others vary widely. As far as I know, we don't do longitudinal studies, but what your results suggest, there could be some dopamine connection with today, I'm slow, and another one is, so I don't know, could you comment on just the stability versus instability of SMT, how that might be related to the story you're telling?
Yeah, that sounds very, very, very interesting. I mean, yeah, it's like a daily dopamine level, right? Or that, who knows, caffeine, some other neurotransmitter might have influenced their motor tapping rate. And it could also possibly affect their cognitive skills, right? Someday you have really good memory, some others. Right. For your participants as well. Exactly. So, yeah, I mean, we have to acknowledge it's just the one shot. We have to finish everything in 20, 25 minutes. But it could very well be that if we do this test-retested paradigm, you may see some variability from some kids. So very, very good point.
So thank you. I'm just wondering, I can't remember if you mentioned this already, what the musical background of the kids was, because we know that music training influences both tapping interval and auditory working memory, and so whether those 2 things are correlated perhaps because of some kind of music training factor.
Yeah, that's a good point. We're actually in the middle of developing good music background questionnaire, but for this study, our question was probably not as precise as what we are developing right now, but that's certainly something that we have to consider down the road. And as I show you, some studies show the connection between rhythm and this working memory skills. So it could very well be that music training can improve their working memory and also influence their tapping rate too.
Sorry, are you listening to us? No. Hello. Hello. We have a question here.
So it's in Argentina.
Hello, thank you for the speak. Was really interesting, and congratulations.
Yeah, hello, Argentina. Do you hear us?
Are you hearing us? Hello? Hello?
Argentina?
Yes. Hello, hello, hello.
Yes. Can you just say hello to us, Argentina?
Hello? 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 Hello, hello, hello?
Okay, yeah, maybe we'll just try a couple more questions in this room.
relationship between tapping and auditory working memory, because basically they're both related to memory but very different, because tapping is a very short time scale, more prediction kind of task, and the task that you show to measure auditory memory is a more explicit longer term task. And there can be like a third factor to explain the relationship, like attention during the task, the experiment itself. So how do you explain the relationship?
Not quite. That's why at the beginning of my talk, I asked how many of you would believe if I measure your finger tapping rate, I can predict your cognitive skill, such as memory. Seemingly very unrelated. But if you look at this neurobiological foundation, which is not showing up here right now, they all based on temporal processing, right? So if you have a faster temporal processing, that will manifest itself as a faster finger tapping rate in the motor domain, but that could also manifest itself as higher working memory ability in the cognitive domain. So they're seemingly very unrelated, but actually when we measure both from within the same participants, we were able to actually predict their working memory score by looking at their rhythm tapping rate in a sort of reverse direction. So kids who tend to tap slowly, right? You have a longer tapping interval. They also have lower working memory score. That's what we found. Does that answer your question? Any other question? We don't have time. Oh, we don't have time. Sorry. So, all right. So let's move on. And I'm gonna. Thank you so much. Okay.
And then we'll.
Well, thank you. Very interested in. Well, thanks so much. Yeah, absolutely.
Hello, La Plata, do you hear me? La Plata.
Hello, hello.
Hey, we hear you now. Wow.
Do you hear me?
I think it's because your microphone was off. Maybe.
No, it's not off.
Okay, well, we're good now. So the next presenter, you're welcome to ask questions. We'll pass the floor over to you. Okay, all right. I'm going to turn this microphone off now. Okay, thank you.
I'm going to introduce him. You said 1 minute, right? Oh, this one. Yeah, yeah.
It's okay. But there's a secondary problem with that, which is the projector.
Mike.
Check. 1, 2. Checking the voice. 1, 2.
Check. Okay, let's move on. So our next speaker is Shinya Fujii from Keio University. He'll be talking about beat synchronization predicts language disturbance in patients with schizophrenia.
Thank you. Thanks, Yune, for a nice introduction. And then thank you for giving us the opportunity to talk about our recent study. So the title of this talk is Beat Synchronization Predicts Language Disturbance in Patients with Schizophrenia. So our central focus is the relationship between the beat synchronization and speech and language functions, such as like the keynote lecture at the noon today. So we are very interested in how these two are correlated with each other, and then if it's have a relationship, why we can see those significant relationships. So previous studies have shown like beat synchronization and language are closely related with each other. So this study by Woodruff Carr et al. Published on the PNAS paper on 2014 has shown that synchronizers showed better phonological awareness and auditory short-term memory, such as Yune said too, and also the rapid naming. So, and then also they measured the EEG measures and then said that maybe the auditory envelope information is well encoded in the synchronizers compared with the non-synchronizers. But we still have to think about why we can see, such as the last discussion, why we can see the, the significant relationship between the language function and the beat synchronization. So I recently published a paper on the frontiers of human neurosciences to propose a model about possible shared brain networks for rhythm processing in music and speech. For example, basal ganglia is very important, such as Jessica Graham has shown. Beat perception is related to the basal ganglia neural activities, and also the connected to the frontal areas, especially the dorsolateral prefrontal cortex, premotor areas, and SMA. And also cerebellum is very important to detect the timing informations. And also the connectivity to the temporal gyrus, that's also very important. So these areas overlapped with each other when we process in the music, the rhythm in music, but also the rhythm in speech too. So right now, I'd like to emphasize the importance of the rhythm processing in clinical situation and setting. So recently, there are a lot of studies about the studying about rhythm processing and Parkinson's disease. And also the, such as like the David Macaulay's group is investigating about the stuttering. And also dyslexia is a very important topic, especially recently. And for example, aphasia too, for example, this Stahl paper has mentioned about not only the pitch factor, but also the rhythm factor is very important to improve from the aphasia. And then also my colleague, Catherine Wang, has shown that proposed auditory motor mapping training is very important for the autism kit. And then now, we are interested in the schizophrenia, but very little is known about the schizophrenia patients. So we are trying to understand how about the beat synchronization ability of the patient with schizophrenia. So here, I'd like to a little bit explain about what is schizophrenia. So schizophrenia is a chronic and severe mental disorder affecting how a person thinks, feels, and behaves. So there are three major symptoms. One of them is positive symptoms, including hallucinations and also delusions. And the second one is negative symptoms, including emotional flatness and extreme apathy. The third one is cognitive impairments, such as like the working memory and also the executive functions. And also, language disturbance is a central feature of schizophrenia and one of the primary behaviors by which it is diagnosed. And a very recent study has also shown that reading is similarly impacted in schizophrenia and dyslexia. So there may be a close relationship with each other between the schizophrenia and the dyslexia. And also, schizophrenia can be viewed as a Parkinson's disease of thought. So that's why basal ganglia is a central focus of the schizophrenia too. So right now, I'd like to explain a little bit about the neural abnormalities in schizophrenia. So this is the very famous study, and then also in the principles of neuroscience. So in the schizophrenia patients, they show a loss of gray matter, and especially around the areas including the dorsolateral prefrontal cortex, and also the temporal areas, like superior temporal gyrus. And then people suggest why loss of gray matter is happening in the schizophrenia. And then another study has shown this may be not the decreased number of the neurons, but more like the decreased dendritic spine density. So this suggests maybe there's some network connecting the areas maybe something wrong with the schizophrenia patients. And then to support these hypotheses, a more recent study is using the tractography. For example, for the language network, arcuate fasciculus, which is the tract fiber connecting the Wernicke and the Broca areas. And then this tractography is very different in the schizophrenia patients compared with the controls. And then especially this study has shown these pathways are related to the auditory verbal hallucinations. Another study, this is also recent, and showing that the front striatal pathways is also very related to the patient with schizophrenia. So here you can see the green areas, which is the frontal areas, and also the orange areas, which is the more like the basal ganglia areas. So these pathways, white matter integrity is reduced in the patient with schizophrenia. So by considering these previous studies, our aims and hypotheses are, our aim is to investigate beat processing abilities and language function in patients with schizophrenia. And then our hypothesis number one is patients with schizophrenia are maybe beat deaf. So we wanted to test this hypothesis. Number two, beat processing ability of patients with schizophrenia predicts the degree of language disturbance. Number three, beat processing ability and of patients with schizophrenia related to frontotemporal or maybe frontostriatal connectivity in the brain. So here, the methods, the participants in our study. So we have two groups, patients with schizophrenia, 45 participants, and healthy controls, 38 participants. So sex and age and years of commencement of musical training and then duration of musical training. So sex is, kind of matched, but age is right now. So this is the preliminary results, so not matched very well. So we are still collecting the data set right now. Years of commencement, very matched, non-significant differences, and then duration of musical training, a slightly more years of training for the healthy controls. So here, I'd like to explain about our task. So first, we use the music tapping test. So we use the musical stimuli, including the three genres of music: rock, pop, orchestra, and jazz. So for example, this musical piece, Heart So Good by John Cougar. So listen to this. So ask the participants to tap with the beat. And also the Boston blocks. And also the jazz music. And also I played these three genres of musical stimuli with the three tempi. So 100 BPM, 120, and 140. And another task is I used was beat interval test from the Harvard beat assessment test. So this Harvard beat assessment, we call this as HBAT. I developed this test with the Gottfried Schrag at Harvard Medical School because we wanted to quantify the individual difference of the beat perception and productions. And then I'm going to introduce a little bit about this task. So this test is kind of questioning the two forced alternative choice tasks. So you have to choose whether the musical stimuli or this metronome stimuli is getting slower or faster. So like this. So this is getting faster, right? Very easy to say. And then this one, the beat is getting slower. It's very easy to say, right? So the thing is, so right now, beat interval is changing very obviously, but parametrically we can change the amount of the change. So I'm going to ask you, how about this metronome stimulus? Getting faster or slower? How many number of people here think it's getting faster? Okay, slower. Oh, very good. It's getting slower, actually. Actually, you know, when I did this in my class, half of the students said it's getting faster. So you guys are really good, I guess, because of the ICMPC people, I guess. So what you guys did was the perception part. So you have to, you know, perceive the change, and you have to answer on the keyboard, getting slower or faster. And then in the H part, you have to also do without saying anything. You can just tap with the stimulus, and then the computer program automatically detect whether your tapping is getting faster or slower. So this is the production part. So without saying anything, just answer by your finger tapping. So we measure the thresholds, and then which millisecond you can detect and say, perceive, and produce the music beat intervals. And then here's methods about the clinical assessment. So we use the positive and negative syndrome scale called PANS to evaluate symptoms of schizophrenia. And then we use the language subscore in the repeatable battery for the assessment of neuropsychological status called Urbans. And then this includes, for example, say name of vegetables and fruits as many as possible in 90 seconds. You can meet with that, like, blah, 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 fruit, orange, banana, etc. And then another one is to answer the name of each pictures like this. What is this? Trumpet. What is this? Chair. What is this? I like this, easy task, right? So we have used this. And also we use the MRI, magnetic resonance imaging, and also diffusion tensor imaging. And then because we are interested in the temporal frontal connectivity and also the striatal frontal connectivities, so we use the DTI imagery to make a tractography. And here's the results. This is the vertical axis of the synchronization index, and then we found that the patients showed worse synchronization compared with the controls. And also, when we analyze the data from the patients, and this is the pop orchestra synchronization index, and a rock song and a jazz song, and then 3 BPMs. And then we did find a significant differences, the effect of the tempo and also the effect of the genres of the music. So patients showed worse synchronization with faster jazz music. And also, this is the typical example of the patient's data. And then the left one is the kind of, this is the circuit plot. And then these are not so synchronized very well. And then this guy is synchronized very well. So we can say the right one is synchronizer, and then left one is non-synchronizer. And then when we look at the language score, left one showed very low language score, and then right one showed the very high language score. So we did a correlation by using the data from the patients, and then this lateral axis is the language score, and the vertical axis is the beat synchronization index, and then we did find a significant correlations between the two measures. And I want to note that this correlation was controlled with the SAS scale, which is the motor scale, general motor scale, and also the chloropromazine dose. So it's the using the medical dose, how much medicine they are taking. So we found worse synchronization, more impairments of language. Here, results from the beat interval test. So the left one is the threshold. So vertical axis show the thresholds. And then left one is a perception result, and then right one is a production result. And then we did find patients have significant difficulty in perceive and produce beat interval changes. So here we further investigated. So vertical axis, so here is a measure from the beat interval perception threshold. So the lateral axis is the tractography result. And then from the left, all acute fasciculus volume. So shown here. So from the Wernicke areas to the Broca areas. So we did find a significant correlation between the two measures. So maybe the perception may be related to the temporal frontal structural connectivity. And this one, yeah, AF tract volume correlated with the beat interval perception. And here, results from the diffusion tensor imaging, but here right now, lateral axis shows the frontal striatal tract volume. So that's shown here from the basal ganglia striatum coded actually, coded areas to the frontal areas. And then, so here the vertical axis is not the perception, but the production thresholds. So we also find a significant correlation between these two measures. The frontal striatal tract correlated with the beat interval production. Okay, so right now, I wanted to find what we did find, summary and discussions. So in this study, I found that patients with schizophrenia showed less beat synchronization than controls. So support our hypothesis that patients with schizophrenia may be beat deaf. But we have to, right now, the preliminary results. So right now, we are still collecting the healthy control data to match the ages and also the expression of the music. And the second, we found degree of beat synchronization was correlated with the degree of language impairment. So this results support our hypothesis that beat processing ability of patients with schizophrenia may predict the degree of language disturbance. And also, we found that patients with schizophrenia showed poor beat interval perception and production abilities than the controls. And then these results suggest that both beat perception and production ability might be impaired. But here, we found that the beat interval perception, so not the production, perception ability was correlated with the tract volume of the arcuate fasciculus. So from the temporal gyrus to send some information to the frontal areas. So we found some correlation between the two measures. And then we also found the beat interval production ability was correlated with the frontal striatal tract volume. So I'm just thinking like, such as what Yune said today, like, the striatal basal ganglia to the frontal area is more related to the production side, such as like the tapping productions. So our results suggest also here, the production ability is related to the striatal frontal connectivities, but more perception side is more related to the STG to, so arcuate fasciculus, so Wernicke to Broca type of, connectivity might be related. So suggesting that dissociated neural pathways for beat perception and productions. Okay. So I'd like to thank our students who involved in this project. Shiori Honda sitting there, and also Ryo Ochi sitting there, and also Aya Kato sitting there too. And then they did a lot of experiments too, and also the scanning, and then also the, did a lot of music tapping tests to the patients. So we appreciate that. And also the our colleagues from the Department of Psychiatry, Mr. Tarumi, Dr. Noda, Dr. Nakajima, and Dr. Mimura, I really thank that. And then lastly, I'd like to mention about the posters. So Shiori Honda and Ryo Ochi gonna make a presentation tomorrow from, I think, 10? Yeah, 10, it's very late. So they're gonna do the poster session. So if you're interested in more, we're gonna show the very detailed results. So please come and let's have a discussion. Okay, thank you very much.
Hello. Thank you for your talk. It was very interesting.
Thanks.
You mentioned that you correlated the activity in the arcuate fasciculus with the beat interval test. Did you correlate it with the beat alignment test?
Not yet. Okay. So we didn't do the beat alignment test, but we did use the musical stimuli used in the beat alignment test too. Mm-hmm. And then we can, still see the, how much correlated with each other, but we haven't done yet, I think.
Okay.
Yeah, but would you expect some correlation between the two? Or?
That was going to be my next question. If
Okay,
yeah.
okay. Yeah, we don't know yet, but might be possible to see that. But music tapping test is more like you hear, you perceive the beat, and also you have to synchronize with that. So not only the perception factors, but also the production factors are related with each other. So I want to see how much the perception scores related to the the synchronization score, and also the production score related to the synchronization score. So by correlating the synchronization measure with those two, perception side and the production side, I think we can see more further discussions. How much we can understand about the language function and the beat synchronization, perception, production, abilities of the patients. Yeah. But thank you for the question. It's very interesting.
No problem. Thank you.
Yeah.
Unless we're not live. I mean, are we around the world right now? Hello, very cool results.
Thank you.
I can't help but wonder, as I'm sure you have too, to what degree does a schizophrenic just kind of bomb any task you give them? So in terms of this, the idea that there might be some kind of causal relationship from beat processing to language processing, or the other way around.
Yeah.
And so I guess I don't know if there's a kind of a music-related or other auditory task. Oh, maybe you talked about it already.
Yeah, I think that's a very nice question. So that's why we did. We have shown that's only the language score here, but we also did a lot of clinical tests too. So including the working memory task and also the executive function type of things, I guess. And also, we thought about that too. And then maybe how much they are taking the medicine, and also the general impact of the getting the schizophrenia and our motor pathways, because taking the medicine, it's the dopamine generic affecting the dopamine system in the brain. So blocking basically the dopamine functions. So it's directly after the motor functions. So we have to control the how much they are taking the medicine. So we control that measures, but still see the correlation between the language function and then beat synchronization.
But I guess what I was getting to is, are there cognitive tasks, musical, otherwise, that schizophrenics perform normally on?
Not normal, I would say.
Okay.
That's why because they are in general have the deficit. So we wanted to dissociate. Is this just doing in general very lower, or it's very specific to the rhythmic function? So that's a challenge actually in this study. So we're trying to separate the things out, but it's difficult right now. I think it's right. Yeah. Okay, please. Hello, Argentina. If there are questions.
Hello, yes, it's a question here.
Thank you very much for your presentation. I'm curious about the melodic content of musical abilities. Did you measure this melodic processing in these patients with schizophrenia? I cannot hear you. You are answering.
Can you hear me? Can you hear me?
Yeah.
Oh, thank you for the question. That's a very excellent question. I really like it. That's my interest too. So right now we are measuring the pitch perception and pitch production ability of the patient with schizophrenia, and also the perception of the chords as well, because previous studies have shown they have something different perception of the consonant and the dissonant sounds. So they cannot discriminate. This is the consonant and dissonant. So we are interested in to measure that aspect too, and how rhythm functions is related to the symptoms, and also the pitch functions is related to the symptoms. So we haven't analyzed yet. So we are trying to see that. But thank you for the questions.
Thank you.
Another questions? If there are, I can take. Yes, could be in this room too. There are. Yes, please.
So I think I saw that the schizophrenic patients had done up to 3 years of musical training.
Yeah.
So how can they have gone through so much musical training and nobody told them that they're beat deaf? Or it's.
I think yeah, duration is 3 years. And then they started, on average 10 years old. And I don't know the self-report. Do you know that? The student tested the patient. They may have the self-report. I guess.
Okay.
And then in general, even though they're healthy patients, even though they report as a beat deaf, if we actually invited to the laboratory and then actually we tested, they're just hesitating to say, like, I'm not so good at beat perception and production. So we, it's a different, like, self-report and actual like performance. So in this case, I guess the patients didn't report, they're beat deaf, I think.
Wait, can I ask another question?
Yeah. If there is no question, yeah, go ahead. I guess.
Yeah, you show that maybe they're split in two kinds, ones who are synchronizers and ones who are not.
Right, I did it, yeah.
Did you do the analysis separate for these two?
Not separate.
Or it's
what do you mean?
Everything is together? The synchronizers and the non-synchronizers.
You mean this one, right?
Yeah. So after that, when you did the correlations, these two groups are collapsed together, right?
The healthy and the controls, you mean? Oh, I used only the patient's data set.
No, but here where you have synchronizers and non-synchronizers.
Yes. So in the patient population, even though it's a patient, some of the patients are really good. Yeah. And then somebody is not so good. And then that's related to the language scores.
So if you plot this if you made this plot in different colors, the synchronizers and the non-synchronizer will be different colors.
Yes.
Would there be two clusters of points, or they'll be mixed?
So the data I'm showing is here and here, I think. So, but cluster, I'm not sure. Here is a cluster, so pretty, I think, not super great, but relatively good. Yeah. Sure. Yeah. All good? Okay. Thank you very much. Oh, thank you very much. Oh, thanks a lot. Okay, here, I trust the microphone.
When we talk with Argentina, your microphone has to turn off.
Okay, I have to do that.
I do that.
Oh, you do that? Okay.
Just so you know.
Yeah, okay.
And do you prefer 20 minutes or 22 minutes for your thoughts? To cut the question or?
Oh yeah, 22 is better. I think. Yeah.
Okay, so
22.
22.
Yeah,
perfect.
Yeah.
Okay.
So and we have. How do I? Oh, okay, I see. And then is this the right? Okay, height.
Yeah.
Okay, and then I have to.
Let's do a check. Let me hear you talk.
Do you hear me? Okay. 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 And I have to put this in back. Okay, and I can use this, right? Yes, sir. Yes. You should move on. Okay. All right. Can you hear me? Yeah. Okay, moving forward, we have next speaker, Jicho Kim from University of Connecticut. And he'll be talking about modeling phase dynamics, relaxation, and multistability in mirror perception. Okay. Thank you for the introduction, and thank you for being here. It's great to be here, to be part of this symposium. Thank you. So I'm going to talk about oscillator models that can explain phase dynamics, relaxation, and multistability in human complex rhythm perception. Here's the overview. First, we're going to discuss signal processing capabilities of canonical oscillator model, paying particular attention to the input coupling function, how linear and nonlinear input coupling function have different signal processing properties. And second, we're using this oscillator model to replicate human tapping data to complex rhythm. And we're going to look closely at phase multistability, relaxation time, and relative phase in the human data. So first, we use the canonical oscillator model. This is a generic mathematical model of oscillation. And as you can see in the equation, its behavior is governed by a differential equation in time. So Z is oscillator state, which is a complex number, so it has both amplitude and phase. And the right-hand side has two parts. First part is the intrinsic part, so which determines the intrinsic behavior of the oscillator. So alpha and beta are intrinsic parameters. Omega is the natural frequency. And the second part, the G, call it G times X, is the input term. So G is the input coupling function being multiplied to external signal X of T. So since the oscillator is a complex number, you can plot it in complex plane. And then here is the oscillator driven by impulse train. So you can see that it's just rotating in the complex plane while driven by. And then on the right, that's the that's the real part of the oscillator shown in time. So when it peaks in the real part peaks, and that's when its oscillator's phase crosses zero. So that's when they fire or they tap, if you will. And we are looking at the effect of having different input coupling function in the oscillator model. For this talk, we are using particular kind of coupling function, which is G, shown here. So basically, it's a function of oscillator phase phi. So shown on the right, the coupling. Oh, sorry about that. Hit the wrong button. So this is coupling phase as a function of oscillator phase. So when rho is 0, then the function is flat as a function of oscillator phase. That means oscillators are sensitive to all input events, no matter where in their phase space the onsets input is falling. But when with positive rho, this coupling function amplitude has a sharp peak near zero phase. That means with this non-zero rho, oscillators are sensitive when they are more sensitive when they're close to zero phase. So, and they can ignore onset events that falling outside their temporal receptive field. So this nonlinear coupling with a positive rho allows selective locking of oscillators, and while they can ignore other onsets, but with linear coupling of flat coupling function, oscillators cannot ignore any onset, so they are driven by all onset events. So let's see what the consequences of this. To show you that, I'm using the simple rhythmic stimuli used in Iverson, Rapp, and Patel. It's called TT0. This is tone silence. Sounds like this. And then as Iverson et al. Showed, this physical stimulus has two possible metric interpretation. You could hear the downbeat on the first onsets, like this, or you can hear onsets on the second events. Right? They're physically the same, but you can have different rhythmic perception. So let's call this first interpretation beat 1 interpretation, and the second one beat 2 interpretation. The question is, can oscillator models explain this perceptual bistability? To test this, we ran 457 oscillators, all tuned to 1.67 hertz. That's that matches the period of the TT0 rhythm, which is 600 milliseconds. And oscillators studied from all different initial conditions. So with nonlinear coupling function, you have about half of oscillators locking to the first onset event, shown in blue, and then there are phase zero crossing shown in the plus with the plus sign, aligns with the first onset events,
whereas the other half of oscillators are locking to the second onset events. So these nonlinear oscillators can model this bistability in human perception, because oscillators can selectively lock into a subset of events. But with linear coupling, all oscillators were drawn to a single state, which with this phase zero crossing actually falling in between onsets, because oscillators cannot ignore any events. They are driven by all of them. So they find a conversion between these two onsets. So these are the initial conditions, set of initial conditions in the complex plane we studied oscillators from. And then the colors are to which steady state these were drawn to. So in blue, shown in blue, are the initial conditions eventually attracted to bit 1 interpretation. And then shown in orange are the initial conditions drawn to bit 2 interpretation. So with nonlinear coupling. But with linear coupling, all oscillators are drawn to this gray area between these two onsets. So, and also if you have good eye, then you can see that, actually if you look at the area near the origin, that's the initial conditions with small amplitude. You see more blue here than orange. But outer here, it's more like 50-50, right? So that shows this is because the oscillators with small initial amplitude are impacted by the first event more. So with small initial amplitude, it's more probable for oscillators to draw to bit 1, which is part of the the first onset event, which is part of bit 1 interpretation. So these are the movies of relative phase of oscillators, but because of this PC mag incompatibility, it plays the movie, but it doesn't play sound, so I'll try to sing it myself. Oh, it does. Okay, I don't have to do that. So with nonlinear coupling, you see that a group of oscillators drawn to bit 1. So this is relative phase to bit 1. So any steady state with 0 phase means it's drawn to bit 1. And the bit 2 is at this angle from bit 1. So you have two groups of oscillators drawn to different states. But with linear coupling, see, all of them are driven by all events. So they have to fall in between those events. So how does this compare with other methods? So if you do FFT of the onset signal for TT0, then and look at the fundamental frequency component, and then that's shown in the purple here. So it just like the oscillator with linear coupling, it straddles between the onsets because what it does is the decomposition of the waveform, not selective locking to certain onset events. And here's the output of a single comb filter whose delay matches the period of this rhythm, 600 milliseconds. So the output peaks are aligned with the onset peaks. But here, the single comb filter output contains both bit 1 and bit 2 responses. So that's not like human tappers or perceivers. They perceive this or that. It's not like you hear both, unless you have schizophrenia. So I don't know, actually. I'm just guessing. So, yeah. So, but oscillators actually drawn to this state or that state. So it's more like individual human, perceiver tappers. So autocorrelation for TT0. So it has all the peaks in peaks at onset periodicity. For example, delta T, that's the shortest IOI. And then capital T, that's the period of the whole rhythm. But it doesn't give you any information about the phase. So there should be some 1.67 hertz or 600 millisecond periodicity. But where's the beat? Where the beat is? It doesn't really tell you. So to conclude the first part, oscillator with nonlinear input coupling function captures multistability in human rhythm perception. And linear and nonlinear input coupling functions have different signal processing properties. So it's not like one is good or one is bad. They're doing different kinds of signal processing. So linear coupling does decomposition of waveform, just like a Fourier transform. But nonlinear coupling allows oscillators to selectively lock to discrete time events. So it's similar to comb filter, but comb filters output tends to have, like all possible responses added together. So we use these models to model the human tapping data. So and that's published in Large Herrera Velasco 2015. And they used rhythms of different complexity levels. So here you see 11 rhythms of 4 different complexity levels. So complexity level 0. This row is level 0 stimulus. Then horizontal axis is time in bits. So it's isochronous. Then level 1, you have just one event of the beat, right? And then this for this one, okay. And then for level 2, you have 2 events of the beat. Level 3, 3. Level 4, you have 4 off-beat events. But since there are 8 events, so there are 4 on-beat events, 4 off-beat events. So then where is the beat, actually? So this is why this level 4 stimuli are called missing pulse rhythm, because if you do the FFT, there's no energy at this beat frequency. Let's so I'm going to play pattern 4. So complex level 4, pattern 1. Can you tap to it? Right. So probably you heard beat 1. You had beat 1 interpretation. Right? Because first event is always strong. Yeah. But it is equally probable if you play this in some other, you start this pattern some other place, or you fade in, then you might hear this too. So they are physically the same, but they're totally different rhythms, right? And here's the summary of human tapping data. So for complex, these are blue dots are circular means of tap phases for each trial. So whenever you see circles gathering around phase of 0, and then that's the people are having beat 1 perception. Then if you look at the complexity level 4, then you can actually see some people tap that beat 1 and some others tap that beat 2. And to summarize this data, you can see that as complexity level increases from left to right, the human response transitions from monostability, just beat 1 response only mostly, to both beat 1 and beat 2, and then there are some people who are not able to tap consistently to this beat frequency. Also, you can from the angle of grand mean average of circular mean, so the relative phase of tapping, mean and relative phase of tapping transitions from negative to positive angle. And then it is not shown in this plot, in this slide, but also it took more time for them to reach steady tapping for complex rhythms compared to simple isochronous rhythm. So we model this with also population oscillators starting from all different initial conditions. They are all tuned to 2 Hertz, and they're driven by these stimuli. So what you see here is a relative phase of oscillators to beat 1. So if you see the phase attracted to 0, then that means it's, oh, sorry, I'm doing this over and over again. Sorry. So 0 phase means beat 1 response. So for complex level 0 and then 1, not shown here, but 2, all oscillators were eventually attracted to beat 1. But with some off-beat events with complex 2, some oscillators took more time until they reached steady state. But level 3, 4, you see both beat 1 and beat 2 responses. For here, beat 1 was 60%, and then 56% of oscillators reached beat 1, others were beat 2. So here is the comparison between human data and model response. So if the model faithfully reflects the transition from a monostability of having just beat 1 response to having bistability of having both beat 1 and beat 2 responses. But interestingly, doesn't really capture, the model didn't capture the inconsistent tapping humans showed. So we'll come back to this point. And here's the comparison of relative phase. So for human tappers, for simple isochronous rhythm, the relative phase was negative, meaning people tapped ahead of stimulus onset. But for as complexity level increases, the people tapped behind. So for actually the relative phase peaking at level 3. And then the model shows the similar trend of increasing relative phase with complexity level. But the model phases were all positive, meaning they were all behind. Probably because they're reactive and they're just driven, not really predicting. Now the comparison of relaxation time. So human tappers, as I mentioned, it took more time, increasingly more time, until they reached steady tapping for more complex rhythms. And you can, as you can see, the model captures this very closely. And interestingly, the relaxation time was longest for complex level 3 for both human and model. The reason is level 4, actually, you have 2 equally strong attractors. So oscillators can be just drawn to whatever the closer attractor without any hesitating. But at complex level 3, actually, you have stronger attractor here and then weaker attractor here. So some oscillators starting closer to the weaker attractor can hesitate before, and finally, before being attracted to a stronger attractor. So that makes the relaxation time for complex level actually longer for complex level 4. So this is the last point here. So we noticed the difference between human and model about the points near the origin. There could be multiple possible answers. So in the human tapping experiment, multiple tempi were used. So maybe human tappers could be stuck at some wrong tempo if the tempi changed a lot between trials. But these oscillators were all tuned to 2 Hz. So it was they didn't really have to find the frequency, but they only need to find the phase. But another possibility is maybe each oscillator state represents a potential interpretation, not the actual perception. So then maybe, and perception may involve competition between multiple possible interpretations. So to test that idea, we made an oscillator population model, which is a system of coupled oscillators driven by common signal. So this system can reach steady state through competition and then reinforcement between oscillators. So the model has 10 coupled oscillators, all tuned to 2 Hz. Now they are starting from random initial conditions. And then we ran I'm showing you three simulations. So first simulation, So you see that all 10 oscillators were drawn to eventually beat 2, but this one was tried to stay at beat 1, but it was pulled off by others because they're all coupled together, so oscillators influence each other. They try to synchronize. Then here, maybe it's 5 versus 5. So 5 oscillators with beat 1 response, another 5 with beat 2, and then here you have all 10 of them drawn to beat 1. So if you plot these states, the mean field of these states, it's complex plane average of these, then you have these 3 different points. Then, so now again, the comparison of human and population model. So different from the individual oscillator model, this coupled oscillator model actually has an attractor near the origin because the oscillators, if they are divided into 2 populations, then their average is actually near 0 because they cancel each other. So we can capture this the attractor in the near the origin with a population model. So here's the second conclusion. So oscillator with nonlinear input coupling can model human tapping to complex rhythms. Especially, we saw that phase multiplicity, relative phase, and relaxation time can be explained by the oscillator models. But these are not really explained by the other method. And then I showed that perceived metrical interpretations may be modeled with population of coupled oscillators instead of individual oscillators. That's it. So I would like to thank my colleagues at Music Dynamics Lab and Oscilloscape, and thank you for your attention.
Thank you very much. It's very interesting results. And then I have a question about your first part of the talk. And then you showed the tatats, the rhythm, have the 2 interpretation of the patterns you can see from the model. But actually, when we can interpret the position of the beat, so there are 3 possible interpretations. So we can actually interpret like ta-ta, 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 right? So in your plot, you can see, you have to see 120 degrees as well. But why you can see only the 2 pattern of the interpretations?
Because these oscillators are just driven. So if they had some internal generator, so if there is another oscillator population that could generate internal driving signal,
so
then So this is just one. So that's why we need actually another layer of oscillators. It could be like auditory, model. And then you could think of this generator as a kind of motor area.
So right now the model is the stimulus-driven type of the model.
Yes. So that's the direction we are taking right now. So you need some intrinsic, oscillation. So these oscillators were just externally driven.
Okay.
So if you add intrinsically driven oscillator, then you can actually have more stable states, as you just pointed out.
Wow, that's pretty cool. Thanks.
Nice talk to you, Chul. Question, so the perceptual multistability stuff where there's a bias between English speakers and Japanese speakers. So the dog, English speakers here, whereas Japanese speakers here, I don't know, tewa. Okay. Do you think you could train this on natural language, just run it on thousands of hours of your nonlinear oscillators and thousands of hours of natural language, and could you produce that bias?
I think that is totally possible, and our model actually do have a learning component. I didn't really this model that I showed you doesn't have any learning component, but we have a Hebbian plasticity actually capable. The models are capable of Hebbian plasticity. So, and then if you have the connections between oscillators, and also we have just one frequency. This is just one frequency population, but if you have multiple frequency population and they're learning their relationship or association between them, probably you could bias one model towards one perception. The other model trained differently could towards another perception. But yeah, so conceptually it's totally possible, but we have to just figure out exactly what kind of plasticity, what kind of the temporal interaction are needed to model that. Yeah, it's a great question. Thank you. La Plata, do you have any question? Hi.
Hi. Guys, do you have a question? No. Okay, we are not experts in this field, so keep going with the questions there, okay? Are you hearing me?
So why 457? It's like a very special number.
No. That was just, you saw the grid of initial conditions, right? So very systematic. So that was not random initial conditions, but the purpose of these simulations were to see all possible behaviors from all possible initial conditions. So that was like from all grid, radial grid of initial conditions. So that's just the number. That from how many angles you're gonna use, how many
depending on how many beams.
Yeah.
Okay.
Exactly.
And so speaking of which, how robust is the model and the qualitative effects to the number of oscillators? If you put many more or much less, what would happen?
I studied these oscillators from this systematic grid of initial conditions. But if you have run an oscillator model, just leave it running, reacting to sound, then you never know where what states they will be in when the stimulus actually hits them. So if you have few oscillators, then maybe they may miss some steady states that are totally possible. But if you have more of them, then it is more probable for them to catch all the steady states. So probably more reliable. Yeah, thank you. Thanks. Oh, thank you. Okay.
Please. You gonna stand or you gonna sit? You're probably gonna stand, right?
Stand, yeah.
You look like a stander. Pardon me?
This slipped out.
Oh yeah, it's just blup blup. It's not the world's best.
I think it's a bit.
Yeah, it should be okay.
Yeah, okay. Cool. All right.
And to your lapel, please. Your presentation is on here, right?
Yeah, they're saying. Oh, does this work?
Okay, like this. Clap. Let's just put it.
Oh, okay. Yeah. Just put it somewhere. Cool. All right.
Tell me about your mom.
My mom is home.
Beautiful. That's good. Just load your presentation.
Sure.
Yeah, you can present the center.
All right. Our last speaker is Federico Visi. His presentation is titled Using Unimanual and Bimanual Tapping to Explore Synchronization with Musical Rhythmic Layers. Take it away.
That's right. All right. Thanks, Yune, for the introduction. So today I'm going to present two studies that we carried out at the University of Hamburg. I did these studies together with two colleagues of mine, David Hammerschmidt and Clemens Wollner. And so before we before I start describing you the studies in detail, I'll just quickly go through a couple of working definitions to provide some background. So in these studies, we look mostly at pulse perception, internal beat perception, which is typically what listeners iterate to as they tap their foot or dance along with the music. And of course, sensory motor synchronization is the coordination of rhythm movement with the external rhythm. So I'm sure you're all familiar with this concept since it's like a rhythm symposium, but you never know. So for these studies, we employ two tapping modalities. The first is unimanual tapping, so tapping with your preferred hand using the index fingers, and the other modality is called alternate bimanual tapping, meaning alternating alternate tapping with left and right index fingers. So the two studies we carried out are designed to study how people perform at different tempi. And so the first study in particular is designed to analyze the performance of unimanual and bimanual tapping to a set of auditory stimuli, which namely are metronomic tracks playing at 16 different tempi. And we asked a group of 30 participants to tap to them in unimanual and alternate bimanual modes. The second experiment instead has a bit more complex stimuli. They're like simple rhythmic buttons with 4 instruments, and it's designed to investigate if the tapping modality has somehow an effect on the perception of the internal beat, and also if the morphology of the rhythmic patterns affects how people interpret the internal beat of the rhythmic patterns. Okay, so there the task is a bit more open. So whereas in the first experiment, the instructions are quite strict. People have to tap 1-1 to the metronome. In the second one, they first listen to the rhythmic track, and then whenever they're ready, they start tapping in time with the music at their preferred rate. So let's go on to experiment 1. And so here is one of the stimulus, which I guess you can a bit expect. So there you go. So people tapped to metronomic tracks at 16 different tempi, and this is a graph that shows the overall results. So what we see here is that, so first of all, red lines, bimanual tapping, blue lines, unimanual tapping. The thick line here is the success rate, so meaning the percentage of successful taps that fall like plus minus a quarter of the inter-onset interval from the actual click time. So here, as you can see, like there's nearly perfect performance on average until around 187 BPM, where both tempi starts declining in terms of success rate, and around the 4 fastest tempi, you start probably getting closer to the biomechanical limit of unimanual tapping, and so unimanual tapping declines significantly faster than bimanual tapping. All right, and the other lines here with the error bars are the absolute synchronous, the mean absolute asynchronous, and the sign synchronous. So there's on average negative asynchrony, and what we can see here is that at slower tempi, the absolute asynchrony is a bit bigger than the the one you find in the middle. And here, of course, there's the fact that you can't have a very big asynchrony, fastest tempo, because the interval is very small. So like in this area here around from 100 to 150, you have like the best performance, because you find both high success rate and low mean asynchronous, all right? So looking at the coefficient of variations of the two tapping modes, Unimanual and Bimanual, basically, as you clearly can see, around from 150 BPM onwards, the tapping sequences become more unstable. There's more variability, and as the tempo increases, this becomes more evident. And at the 4 fastest tempi, there's also like big error bars, so like lots of variability in performance. So we carried out a pair sample t-test for the 4 fastest tempi to see if there's a significant differences in terms of performance in the 2 tapping modes. And so what we found out is that the mean coefficient of variation in bimanual mode is lower than that one in unimanual mode, meaning that in bimanual mode the tapping sequences are more stable, and also that the mean success rate is higher. And so like both differences are significant, which tells us that at fast tempi there's a significantly better performance. And so to move on to experiment 2, so here, as I anticipated, the task was a bit more open. Participants were presented with 24 different musical patterns and were asked to listen to them and start tapping at their preferred rate whenever they were ready. So this is like the bass pattern. So it's a what Simon Henriksen called a pump shack pattern, or also known as 4 on the floor. So like a kick drum pounding on the quarter note, snare drum on a half note, hi-hat 8 note, and there's a ascending bass line that plays like the first 4 degrees of a minor scale, but like this. You can tap if you want, or dance too, for that matter. All right. So this is the bass pattern, and from this pattern we created 24 permutations in total by simply assigning each instrument to a different rhythmic subdivision every time. So like this is the bass pattern that I just played. There's the bass on the whole note, snare on the half note, kick on the quarter note, hi-hat on the 8 note, and then by permutating the instruments, you get new patterns such as this one. Relatively similar, it's just a swap there in the middle. And another example, here you get the kick drum with the 8 notes. So. All right, last one. So same material, but swapped in terms of rhythmic layers. All right. Okay, cool. Let's have a look at some results. So first of all, here we're like these bar charts show the number of synchronizations per rhythmic layer in unimanual mode and bimanual mode. So as you can see right away, the great majority of synchronizations are on the quarter note level. In both modes. But what's interesting here, and it's something that we actually investigated further, is that as you can see, it looks as if there's a shift towards faster rhythmic subdivision in bimanual mode, because there are like less synchronizations with the half-knot subdivisions, a lot more with the 8-knot subdivision, a bit less with the quarter note. So to verify this, we carried out an ANOVA on the mean intertap interval, and basically it showed us that like in bimanual mode, people tend to synchronize with faster rhythmic, not tend, but it's like the difference significant. So they synchronize with faster rhythmic subdivision. And there is no significant effect of laterality or handedness. And in this case, musical training didn't have any significant effect. And so we interpreted this result as a action-based effect on internal beat perception. This perhaps resonates with an active account of music cognitions, meaning that the way you move to music affect the way you perceive it. So like here, the task that they were asked to perform somehow had an effect on the internal beat perception. Okay, so still on experiment 2, we also perform multidimensional scaling of the 24 musical patterns. This was done in order to have a distance measure that basically indicated the difference between the base pattern, which is this red dot here, and all the other 24. So these dashed lines are the Euclidean distances between the base pattern, each one of the other 23 patterns in this ordinal space here that we created through multidimensional scaling. And so if we go back to the stimuli, you can hear that this is rather close to the 4-on-the-floor pattern of the bass pattern, whereas number 7 there are like more permutations, so like the difference is larger. And 21 up there, lots of permutations, so the distance is actually bigger. Okay. So what we wanted to do here was to correlate this distance measure with another measurement that we got from the data, which is the coefficient of unlikability, which perhaps not everyone is familiar with. It's a measure of categorical variability. So it tells you how much a categorical variable, like it's a measure of spread of a categorical variable, basically. And so in the case of this study, the coefficient of unlikability basically indicates how similarly participants synchronized to each pattern, all right? So, for example, if nearly all the participants prefer tapping to the same rhythmic subdivision, the value will be low, whereas reaching 0 if all the participants synchronize to the same subdivisions. Whereas if the distribution is more spread more evenly, the value of the coefficient of unlikability will be higher. So we correlated that distance measure of the multidimensional scaling with the coefficient of unlikability, and it turns out that in both modes they're positively and significantly correlated. And so this is, we interpreted this result like, I think this suggests that there is more consensus in how to synchronize to highly codified rhythmic patterns such as the 4 to the floor. And whereas patterns that are further away from those highly codified, rhythm instead result in synchronization that are more spread. So there is, so to speak, less consensus in how to synchronize to those rhythms, because perhaps they're less codified, or they're less there are less strong cultural connotations. All right, and so this is another correlation we ran involving the mean coefficient of variation of the first experiment and the standard deviation of the intertap interval of the second experiment. And so we wanted to see if there was some correlation between how people performed in the first experiment in terms of tapping stability and the consistency they had in the second experiment in terms of synchronization with a rhythmic layer. Okay, and so here we can see that there's like a significant correlation for bimanual mode and just like a trend in unimanual mode, and so we interpreted this trend like this trend perhaps suggests that people that have a more stable performance are more consistent in like finding the internal bit in the other parts. So there's perhaps this correlation between higher, like the interpretation of hierarchical features of the patterns in the second experiment, and more consistent tapping with lower hierarchical metrical levels in the first one. Okay, thanks. So to summarize the takeaways of these two experiments, so the most stable tapping sequences that we found on average were between 83, so the lower coefficient of variations of the tapping sequences are found on average between 83 and 100 BPMs. And so at fast tempi, bimanual mode yields more stable tapping sequences, as shown by the results of the ANOVA and the other slides. And so, yeah, this other result, participants with more years of musical training significantly perform better. I didn't include the slides about that because of time constraints, but this will be described in detail in a hopefully upcoming journal article. And so, yeah, internal beat perception is influenced by how synchronization is physically performed. So there's this action-based effect on internal beat perception. And so, yeah, this is previous slides. Participants that had more consistent interpretation of the internal beat tended to perform more stably in the first experiment. And finally, to conclude, the significant correlations between that distance measure from the base pattern and the coefficient of unlikability perhaps suggests that participants agreed more on which rhythmic subdivisions to synchronize with to patterns that are more similar with highly codified musical structures. And so this perhaps also points to cultural factors affecting pulse recognitions and synchronizations. And time's up. So thank you and merci.
Hi, very interesting talk. In light of our earlier discussion about auditory working memory and tapping, I wanted to see if you had any data about auditory working memory and how that might correlate with this data.
Oh, auditory working memory. I'm afraid no, we don't have that, to be honest with you. But it would be interesting, definitely interesting to see. Yeah, that, absolutely. Especially in the second part, perhaps that finding regarding highly codified rhythms may have some, yeah, there could be something there in terms of also auditory working memories, but of course we don't have the data to see that.
I very much enjoyed that. Based on your sort of closing thing, looking at this sort of cultural impact potentially here, in your experiment too, did you see any difference in overall performing? So like on the CV measures, based on the voicing of the quarter note? So did you see better tapping if the bass happened to be the instrument doing the quarter note?
Okay, that's there. We've looked at various things. So we didn't check the. Yeah, that's an interesting thing. We didn't check that particular thing you mentioned. We looked at, for example, we carried out a few chi-square tests to see if there was an effect of the instruments that played at the quarter note level, for example, which is maybe something you might be interested in. And it turns out that for just the quarter note level, where the highest number of synchronizations were found, there was no significant effect of the instruments, whereas in the other ones, there was, for example, the snare drum was more prominent. People tended to tap more often to the snare drum compared to the hi-hat, which had fewer synchronizations. Yeah.
Hi, La Plata. Do you have any questions? La Plata.
Hi. Yeah, hi. Do you have any questions? No? I have a question. Do you think that maybe there are some acoustical or sound or musical features that are affording the different tappings, specifically in the experiment too?
Okay, yeah. We've done, as I was telling the person that asked the previous question, we had a look at how the instruments influence the synchronizations. We haven't done an analysis on the acoustical features of the patterns. That could be something that we could do in the future. Regarding the instrument that plays at each subdivisions, as I mentioned earlier, there's an effect there. Like these people tended to prefer synchronizations with the snare drum and the bass drum, whereas the hi-hat didn't receive as many synchronizations as the other instruments. But indeed, we could also look at other acoustic features of the patterns and see if there's something there.
Okay, thank you.
Thank you for your question.
Thank you, La Plata.
Following up on La Plata's question, this was sort of mine, and a suggestion is it's been found that certain sonic features, musical features, especially low frequency spectral flux in the 100 and 200 hertz band, are strong predictors of beat salience. And the interesting thing there is you don't look at individual instruments, but you look at the total audio signal. So you do an audio signal analysis.
Sure.
So when you speed up the bass part, for example, you get a lot more high frequency information.
Yeah.
So it's not necessarily a low frequency cue, but that would be my first suggestion as to where you might see the correlations. And it would be interesting to see where people who might have done the difference between bimanual and unimanual tapping, where you've got higher amounts of low frequency flux, affords bimanual tapping more because you want to use both hands. So that would be I'm wondering if you think that might be an interesting way to look at what you've got.
Oh yeah, absolutely. Thanks for the suggestion. Yeah, I totally agree. Yeah. Thank you. Yeah, yeah, that's nice, no?
Have you looked at any a musical background of the participants? Because I play the drums and then I can imagine for some of the patterns in the experiment too, it's very easy to play, but some of the patterns is I can imagine it's difficult to play. So in terms of the like motor constraints or motor imagery, there is a pattern. It is very easy to imagine to tap with the beat, but some of the pattern is very, not so familiar with it. So if you are, surveying some background, participants, I'm just wondering whether there is some effect of that.
Yeah. Well, thanks for your question. Actually, we did because we collected also demographic data also regarding musical background of each one of the participants. There were 30. And so what we tried to look at, we basically flagged each one of the participants that played a rhythmic musical instrument such as drums or marimba, etc., and try to see if that person if there was anything, basically. Unfortunately, there weren't many, so we didn't have enough, I think, because there were just, I think, 3 or 4 participants that played drums or pitched percussion or something like that. So yeah, we didn't find anything significant. So I suspect we would have to collect more data, basically. Yeah, well, that would be interesting, especially if they play in the same band.
Okay, thanks everyone. We're ending the session. Thanks for coming to our RMB session. And this is it.
da da